Okay, thank you.
Thank you.
Anyways,
okay, let's wait
I
for-
hope it works.
Yeah, it will work.
Thank you.
Thank you.
Young Chun, are you going to introduce me?
Yes. Can you hear me?
Yes.
Okay, great. Let's wait for Yak.
Pardon?
Let's wait for Yak.
Oh, okay, sure.
Last common ancestor to whom? Fish?
To me.
Well, that would be your brother or sister or your mother and father.
No, no, no. My last common ancestor goes much deeper than my parents.
Well, maybe it goes back to some microbes somewhere.
It goes deeper than that.
Okay, okay. Well, I was just wondering.
You should, that's great. I mean, the more wonder, the better. I shared that common ancestor with you, Andrew.
Yes. And with various bugs.
It's not bugs.
Okay. The division seminar next Monday has a former student, Tanya Bosak, talking about how microbes become fossils.
Oh, that's great. That's unbelievably important
Yeah.
because of the micropaleontology that was begun with Barghorn, especially stromatolites and banded irons and stuff.
Right. She has a stromatolite laboratory that actively makes stromatolites.
How interesting. Who's going to give the talk?
A professor of, I don't know, geology in at MIT named.
Oh, I know her.
Tanya.
I know who she is.
Yeah. And she used to be a
She's
student.
great.
Here.
She used to be here with in the microbiology department here
That's right.
with Diane.
Yep. She and I wrote a good paper, but She was interested in, I think she had a better topic for a thesis than mine.
Uh-huh. I'll certainly be there.
Hello, Hyman. I am sorry, Sissy, we were late from our class, and as usual, so you missed a great discussion from the class on the definition of life.
Okay. I couldn't make the class because I had to prepare the talk here with Yang Cheng. The answer I'm going to give them will be the answer what life is all about.
Very good, very good. Yeah, we have invited people from the class to hear the.
Okay, are we ready?
So Yang Chen, why don't you start?
Okay. Hello, everybody. Today, our speaker is Dr. Hyman Hartman. Dr. Hartman graduated with a Bachelor of Science degree in biochemistry from McGill University in 1957 and got his PhD degree in biochemistry at Columbia University in 1963. After that, he has worked at MIT, UC Berkeley, the University of Tel Aviv, and Harvard Medical School. He is now a research scientist in the Department of Biology at MIT and the director of the Institute for Advanced Studies in Biology at Berkeley, California. Dr. Hartman's research focused on the origin of life. He has published many books on this topic, including Search for the Universal Ancestor in 1987, Clay Minerals and the Origin of Life in 1986, and The Origin of Evolution of the Cell in 1992. Today, he will talk about Darwin and the Tree of Life. This is the second talk of his 3 life seminar talks. Please also come next week for his last talk. Let's welcome our speaker.
So thank you. I am humbled. Today, last week, I talked about Aristotle and embryology and talked about the formal cause and the material cause, etc. This week, I am going to essentially do the same thing, except that now it's Darwin who is the master that I must follow. Now, Darwin and the last common ancestor is the nature of this talk. So I'll give you a little preview. Darwin's tree of life, it all has to do with the tree of life. Haeckel's tree of life applied to embryology. The biogenetic law that ontogeny recapitulates phylogeny, which is really another way of saying terminal addition. Catastrophe and evolution, the origin of the eukaryotic cell, the Woe's tree, the genetic code, metabolism, and photosynthesis. That's what I'm going to be talking about. So let's begin. Here is the man who changed biology completely. And he is the man who wrote the famous book on the origin of species in 1859. And here is the only diagram in the whole book of the origin of species. At the bottom, A, B, C, D, E, F, G, H, I, there's no J, K, L, are these original species. Each level is about 100,000 or 10,000 generations or something. And what you see is that A is gradually bifurcating into a series of evolution and ending up with 3 series at the top, at not the very top, but at the 10, at the number 10 top. I also does that. Then there's a number of species that die. They just go up and then they disappear. And so you have now 2 things, the species are increasing, some of them. And now the question is, what's going on? So this was a fellow named Kuchera who put it on the 5 sub-theories which Ernst Mayr said that Darwin had. One is the descent with modification. This is that the idea that species change and give rise to new species, and they share a common ancestor. That is the most important point. That is, with Darwin, you go down the tree and you end up with the common ancestor. Then the other point that Darwin believed was that species evolved very slowly. There were no big breaks. The other one was that some species multiply. And the 5th one was natural and sexual selection. Natural selection, as you all know, is that there's more progeny and sexually recombining populations than is adequate for the natural resources. So therefore there's competition, and then the ones that are more favorable go on and form the next generation. And sexual selection had to do with the fact that females select their males, and as a result of it, you have sexual selection. So this is what Darwin showed, and this was on his tree. And Darwin then, great, and said, well, let's put it to all of life. The affinities of all beings of the same class have sometimes been represented by a great tree. This simile speaks the truth. Green and budding twigs may represent existing species, and the other ones are those that produced during former years may represent the long succession of extinct species. And then at the very bottom, he says, you know, buds give rise to fresh buds, and vigorous branch out and overtall all the other ones. And so by generation, I believe it's been with the great tree of life, which fills the earth, the crust of the earth, with dead organisms, basically, and covers the surface with ever-branching ramifications. The fact that there's a lot of evidence in the crust of the earth with the broken branches is, of course, paleontology. Now I want to introduce you to the man who I'm going to talk about, and he is Ernst Haeckel. He was a German zoologist, a naturalist, and a eugenicist, and a philosopher, a physician, a marine, a professor, marine biologist, and artist who discovered, described, and named thousands of new species, especially in the phytoplankton, and mapped a genealogical tree relating all life forms and coined many terms in biology, including ecology, phylum, phylogeny, and protista, which are single-celled eukaryotic cells. He was called the German Darwin as he promoted and popularized Charles Darwin's work in Germany. He not only identified the amniota, but he's best known for his biogenetic law, ontogeny recapitulates phylogeny. Haeckel developed the recapitulating theory that ontogeny recapitulates phylogeny, claiming that an individual's organism's biological development, or ontogeny, or embryology, parallels and summarizes its species evolutionary development, or phylogeny. So I'm going to go back to my embryology and see how far I can go down. But before I do that, Haeckel had a person at the University of Jena who was a linguist. And so Haeckel gave Darwin's book on the origin of species to this guy Schleicher. And Schleicher said, Well, there's nothing in Darwin that I don't know, because the tree that he built in the only diagram in the Origin of Species, I've been drawing for years on the evolution of languages. And of course, here is the Romance languages, of course, which come out of Latin. And then there's Germanic languages. And then there's Slavonic languages like Russian, etc. German languages like German, and also a lot of English. And so he said, and this is a very, very interesting statement, but for the invention of the art of writing, the student of language would never have imagined up to the present day that such languages as Russian, German, and French, for example, are descended, after all, from one and the same stock. Nay, it is quite possible that no one would have ever hit upon the idea of a common origin for any languages whatsoever, however closely related, or ever would have supposed that language is subjected to change at all. Now, this is, I would say, a very, very interesting, how should I say it, foreseeing of what was going to happen with molecular biology, because that finally gave us a written language for which we can now reconstruct our phylogeny and our evolution. It's just and Darwin and the evolution of language dominated the 19th century. And here's the book, Language, Race, and Natural Theology in the 19th Century. Okay, here is the tree that was stimulated by the language tree and, of course, Darwin's tree. And here is the tree of Haeckel, Haeckel's tree. It is a fantastic achievement. Up here in this little round place here is the amniota. And in the animalia part of the tree, and right at the top is the amniota. Now, let us recall what the amniotas are. The amniotas are the reptiles, the birds, and the mammals. And they, as you go down the tree, came from an amniota, which is something that branched into two parts. One branch of the amniota was going to evolve into the mammals. The other branch of the amniota was going to evolve into the reptiles. And the reptiles were going to evolve eventually into birds. Then below the amniota are the amphibians, things like frogs and salamanders. And then below them are the fish. And below them are this group of organisms called, that is represented by Amphioxus. So going down this tree, we end up with Amphioxus. Well, then first, let's go to the amniota for a second. The amniota were first described by Haeckel and on the basis of the amnion. And this is the formation of a membrane which distinguishes reptiles, birds, and mammals from other vertebrata. It's evidently a very important process in their ontogeny and in their phylogeny, which corresponds with it. It coincides with a series of other processes which is essentially determine the higher development of the amniota. Now, if you recall my lecture last week, the first 2 weeks of as an embryo was related to the ontogeny of the amniotic egg. And only after that did I begin to develop as an embryo. Now, here is what the amniotic egg first arose. It arose in the reptiles. And here you find the outer shell, of course, and then there's the chorion, which is a membrane that surrounds it. And then there's the egg yolk, and then there's the allantoa and the amnion. This defines the egg. Now, why is this egg of such great importance? Because this egg evolved out of an amphibian egg. And the amphibian egg originally had to do with being in water. It had to be in water. And this egg allowed the reptiles to now take over the land. That means the land animals now resulted because of this egg. And you wonder, is there such an egg that you have noticed recently? And the answer is yes. At breakfast, hopefully the chicken wasn't in there, but in general, here are the four membranes. There's the yolk sac, there's the chorion, there's the allantoe, and there's the amnion. This is the egg, the bird egg. Now, in us and in the marsupials, they're oviparous. We don't lay eggs. The reptiles do and the birds do. And therefore, they have converted the yolk sac and the allantoa into placenta. In the marsupials, they converted the yolk sac into the placenta. And in us and in the mammals, in the placentals, they converted the chorion and the allantoa. And here is how I looked in the 3rd week after fertilization. Here, most of that time is spent in building the egg, in building the egg into the uterus. And up here in this little region between the yolk and the amnion is the embryo. And here is how the embryo develops. Just here, it points out, here's the yolk sac, amnion, here's the allantoe, and here's the embryo. The embryo starts off as a small plaque of 2 layers, ectoderm and endoderm. Then comes this primitive streak, and then that is gastrulation. Gastrulation is the most important point in vertebrate evolution. And that is the point where the layers become 3 layers. They become the ectoderm, the mesoderm, and the endoderm. And then from this gastrulation, you get the neurulation, and gradually you end up with the pharyngeal system on day 25. That means 4 weeks, between 3 and 4 weeks, all of this takes place. And this is what happens in the 4th week, and this is the human embryo, and this is the embryo that Aristotle began his studies on, if you recall. And here's an amazing fact about this embryo. This is the embryo of 4 weeks after embedding into the uterus, and this is the embryo after 4 weeks. This is the 96-hour chick embryo that we discussed with Aristotle. And here you see there is this enormous similarity between this and this. Now, why is this of great importance? Because Darwin, in his book, said, holy moly, that you look at the embryo of various organisms, and then you find out that in general, the embryos of the most distinct belong are similar and then become fully developed. A better proof of this was the statement by von Baer that the embryos of mammalia, birds, lizards, and snakes, this is of course the amniota, and chelone, which is a turtle, are in the earliest stages exceeding like one another, both as a whole and as a development of their parts. So much, in fact, we can distinguish embryos only by their, I can't see the rest of the thing. You have to read it. In my possession are two little embryos in spirit. This is von Baer, the greatest embryologist after Aristotle, in spirit, whose names I've committed to admit, and I'm unable to say what class they belong. They may be lizards or small birds or very young mammals. The similarity of the head, etc., everything, even if they existed in the earliest stage, we would have known nothing for the feet, etc., etc., etc. And the feet all arise from the same fundamental form. So therefore, what you saw in the fourth week of my embryology was the form that was now going to dominate all of the amniotic system. And this is a very famous picture by Haeckel, who immediately took up this fact that the embryo, now look at the top and then go down each one in the column. Now at the top is the so-called pharyngeal system. That was what we saw at the end of week 3, somewhere between week 3 and week 4. Now look at what happens in the fish. The fish does not really differ very much from the pharyngeal system. The amphibian is also very similar as you go down. The chick and the turtle, you can see, go into what we would call the fourth day of my fourth week of my embryogenesis, or the fourth day of the chick or the turtle. And of now, to just look at these things in terms of their names, this going down the thing is the fish, going down here is the salamander, going down here is the tortoise, going down here is the chick, hog, calf, rabbit, human. But the stage of the fourth day of the chick and the fourth week of my thing are all extremely similar. And as you can, here's the reminder of what happened in the third and early fourth week. Sorry. Now, there are two models that have to do with this system. One is called the hourglass model. The other one is called the just going up straight. And it all has to do with what happens with the fertilized cell, which is we would call the egg cell, which then goes on to form a multicellular system, and then blastula, and then gastrula, and then planula, neurula. Anyways, here's what is the simple form, where the egg starts and it multiplies, forms the blastula, blastocell, and then it curves in on itself and becomes the gastrula. Now, from these identical gastrulations of representatives of most different animal phyla, from porphorians, which is these are simple simple organisms, to vertebrates, I include, according to the biogenetic law, that the animal phyla have a common descent from one unique ancestor, which in essence was identical to the gastrula, and it's called the gastraea. And this is the 4-week one. So you see that what is happening is that you start off with a fertilized egg cell, which forms a blastula. And in us, it goes through a number of steps before you get to gastrulation. Then it forms this frangilla, which I showed you. And then in the 4th week, it becomes a human embryo. And this is ontogeny recapitulating phylogeny. And it shows you that in some sense, you are related to all these other animals. You're an animal, you're a vertebrate, and you're a mammal, and then a primate, and then a humanoid system, and then a homo, and then finally sapiens. So this does not mean that ontogeny recapitulates phylogeny. All it means is that in our embryogenesis, we are all coming from a common root. So let's go back to Haeckel's vertebrate tree. We have gone down from the amniota through the amphibians into and the fish, because all of these are related to the gastraea, to the amphioxus. And this is the amphioxus. And Haeckel filled the first Darwinistic tree in his book, General Morphology, with cosmic ambitions, new technical terms, ferocious polemics, bold pedigrees, all the way up to human beings and taxonic revisions, which promoted amphioxus to a new subphylum, the skulless ones. Haeckel believed that the lancet, which is the same thing as the amphioxus, was the last of the Mohicans, representing a lower class of vertebrate that had largely vanished from the fossil record according to the absence of a solid skeleton. His high esteem for the lancet was evident in the book, The Evolution of Man, 1874, where he admonished his readers to regard lancets with special veneration as the animal which alone of all extant animals can enable us to form an approximate conception of our earliest Silurian vertebrate ancestors. The lancet was now central to Haeckel's theory that ontogeny recapitulates phylogeny. In reaction to Haeckel's enthusiasm, a church newspaper complained that reverence for the lancet caused the dignity of humanity to be trodden underfoot and the divine rational conscience of man to be grievously hurt. In 1874, you should read it yourself because I can't.
And what was the punishment for committing such a sin?
Nothing. There was no punishment.
No punishment from the divine authorities.
Anyways, you should read what it says. I can't read the full thing because of it's somewhat blocked by the people who are on here. But read the full thing, just take a few minutes because it's very amusing. And there was a song. It's a long way from amphioxus. And the song goes, it's a long way from amphioxus. It's a long way to us. It's a long way from amphioxus to the meanest human cuss. It's goodbye, finnan and gill slits, welcome skin and hair. Now, if I recall the time with Aristotle, there was the notion of that the species was immortal and that there was an archetype, which is the form. And now what happened with Darwin is that the archetype now was becoming the ancestor. So I'll just give you a few skill. Here is Owen, who is a famous fellow who argued vociferously against Darwin. Here is his archetype, but now the archetype is something that's occurring in the vertebrate, in a subphylum. So it's not the archetype of human, it's the archetype of the phylum, of the subphylum. So here is the archetype. There's the fish. There's probably a reptile. This may be a mammal. And then there's the ostrich, a bird. And then there's the magnificent human being. And here is the archetype of the subphylum. And here is Amphioxus. And it now, this is a recent paper, Amphioxus genome illuminates vertebrate origins and cephalochordate biology. Haeckel was right. Here is a, how shall I say it? A fossil found in the Burgess Shale that's 550 million years old. And when they saw this, they said, this is Amphioxus. Now I want to go back to the origin of, this has to do with Amphioxus and embryology. So the Amphioxus of embryology goes back and forms the gastria. This is the gastria. Gastrulation of Amphioxus, 3 stages in the formation of the blastula, the curving of the thing, and the complete gastrula segmentation in primitive gut cavity. And here's what he was saying in the embryology of the Amphioxus and the gastrulation. But now, let's go back, and this is Wolpert on gastrulation, and this is his paper on gastrulation and the evolution of development. The origin of the gastrula can be accounted for in terms of Haeckel's gastria theory and early metazoan representing the gastrula. The biogenetic law and gastria theory. This is another paper from Haeckel's discoveries to contemporary views. The biogenetic law of Haeckel is ontogeny recapitulates phylogeny. It only works, by the way, if the following thing is true. Here is ontogeny on the bottom, which is the embryogenesis. In the vertical axis is the species and its evolution in that axis. And as you can see, it is the terminal additions to that phylogeny, which is respected in the ontogeny of, so looking at my embryology through the eyes of Haeckel, I started off as a fertilized egg and then eventuated into a gastrula, which then went through the stages of neurulation and the formation of the pharyngia. And then I went into the 4-week-old embryo, which was an embryo that underlay all the amniotes. Ontogeny recapitulates phylogeny.
Can I interrupt?
Yeah.
With a question. Certainly in details you can have examples where several species or several phyla or whatever families have developed the same trait. In other words, the same parents,
Are you talking
the
about
same limbs
convergence?
on the trees. Yeah, okay, just leave it at that. And that is not the same as this diagram.
I didn't say that. All I'm saying is that my embryology tracked through the tree gives you this terminal addition, that's all.
But maybe that's not true. Maybe maybe life.
Anyway, look, I'm not going to get into this thing right now because that's a whole other question. I don't understand it. But let me now just go further because I can't stop now.
Okay.
Ontogeny recapitulates phylogeny, and that's not me, that's everybody else in the field who studied this particular ontogeny and this particular phylogeny. Now I want to say something about paleontology, because paleontology is another record of what happened in the past. And Haeckel drew another tree. And this tree is called the paleontological tree. And what you see here is the evolution from something down here like the Amphioxus, then through the age of fishes, which then goes through the age of amphibia, then goes through the age of reptiles, and then goes through the age of mammals. And I want you to look at what happens at the Cretaceous versus the so-called tertiary period. It's also the Cenozoic period. And there is an enormous loss in reptiles. Only a small group of things go through. One of them is the birds, and the other ones are snake and lizards and crocodiles. And what happened at the Cretaceous tertiary boundary? So it turns out that I had some interest in that area because Luis Alvarez and Walter Alvarez discovered a ingubio between two calcareous rock formations, a clay layer which was very enriched in iridium. And the question was, what happened? Iridium is a very rare entity in the crust of the Earth, but it is very, very prominent in bolids, in carbonaceous chondrites, and in bolids that come in from outer space. So this was immediately interpreted as, wow, this was a bolid that hit the Earth, and that was the end of the dinosaurs. Now, the forams that built that carcineous group of the carcineous rocks, above it in the tertiary and below it in the Cretaceous, are these foraminifera. And these are single cells that have these very complex calcareous exoskeletons. And they build up these huge rocks as they fall down in probably oceans of some sort. Then there's another group of plankton that are siliceous. Their exoskeletons are built out of silica. Now, here is the big event. The calcareous organisms got wiped out. That's why there was a clay layer between these two rock formations. So therefore, and the diatomic rocks went right through. So this led us to the acid rain hypothesis. So the violent cometary or asteroidal impact events comparable to that which terminated the Cretaceous era must produce large amounts of nitric oxide sufficient to provide NOx mole fractions much larger than 100 ppm over areas of millions of square kilometers. Rapid production of NO2 and subsequent rainout of nitric acid and other strong acids can acidify surface waters, selectively destroying calcium carbonate shells. This scenario holds the promise of explaining both the scope of Cretaceous biological extinction and their extraordinary selectivity. The answer came that here at Caltech, Ahrens in the geology department determined, after they determined where the bolide hit at Chicxulub, that it was more likely sulfuric acid rather than nitric acid. And so there is a interesting interaction that we had with Caltech. The second point I wanted to make about, now this means that at these bolide things, big changes occurred in the age of the dominant species or the dominant organisms. So you went from fish to amphibians to reptiles to mammals. And so there is a real interesting story to be told about what happens at these catastrophic events. What does it do to evolution? And that's something that we are now thinking about and worrying about. The next point I wanted to make was, if you recall, that it was micropaleontology that was dominant in the problem of the Carbonaceous, in the term of the Cretaceous tertiary boundary. And so there was a development by Elso Barghorn at Harvard, and he was a paleobotanist called by his student, Andy Knoll, as the father of Precambrian paleontology. And there were two students that went on, Andy Knoll and J. William Schaff. And they did a fantastic job of looking at now microbial and single cell evolution based on things like stromatolites and banded iron formations. And I'm not going to go into that, but those are really interesting studies. So now I'm back to the one cell. If I go down the tree of Haeckel, I end up here at this juncture before the cell that created the gastria and all the rest of the vertebrates, etc., and the multicellular animals, and also the protista, which are single cell eukaryotes, and plants, which are multi-celled eukaryotes. That it is now to where I am, to my real ancestor. This is what is the ancestor of me and everybody else, I hope, listening to me still. So the interesting point is, I want to just point out the interesting questions. There's the nucleus, there is these two central bodies, there is the chloroplast and the mitochondria. Then there's these other things around here, but the cell is now, this is the cell that gave rise to all of us. And the man who has the best book on the subject is Edmund Wilson, Ellen B. Wilson, The Cell in Heredity and in Development and Heredity. Every buddy who's studying us and the cell should read that book. It's a masterpiece. Okay, that is the cell that Lynn Sagan began to discuss on the origin of the mitosing cells. The theory of the origin of eukaryotic cells, cells which divide by classical mitosis, is presented. By hypothesis, three fundamental organelles, the mitochondria, the photosynthetic plasmids, and the 9+2 basal bodies of flagella are themselves once free living prokaryotic cells. So the eukaryotic cell is not something that came out of a single cell. It is an endosymbiotic system in which cells came together and formed a higher cell. Now, we should recall that this is the cell that gave rise to us after the sperm gave its nucleus to the egg. And so what happened? This sperm and egg, in the nucleus of the sperm and in the nucleus of the egg are the chromosomes. And there are 23 chromosomes in the sperm and 23 chromosomes in the egg. And there's a special one in the 23rd one, which is that if you have an X chromosome from the mother, which always is there, and an X chromosome from the sperm, you will become a female. If you have an X chromosome from the mother and a Y chromosome from the male, you will become a male. These are the sex chromosomes. The other ones are called autosomes, and they essentially are each one is one is from the mother, one is from the father, and they are very closely related to each other. Now, I have to make a bit of an excuse that there is a chapter that came between Darwin and what I'm now talking about, and that is the chapter about Mendel and the history of heredity, because Darwin had one huge lack. He did not have a good theory of memory and history because of inheritance. So this is now just a very short statement. Humans have between 20,000 and 25,000 genes. That means a segment of DNA that codes for a protein. Every person has 2 copies of each gene, one inherited from each parent. The Drosophila chromosome is estimated to have approximately 13,000 genes as compared to 20,000 in humans. Human genome is 3 billion nucleotides and in sequences. That's the human genome. The Drosophila genome is 150 million nucleotides in its genome. The question is, why does less than 2% of the DNA in our chromosomes code for proteins? That is a question that I'd like to answer. Here's what happens in mitosis. Those 2 little bodies up there divide and separate, and then they go to the poles. And here is like a model of just 2 chromosomes. The chromosomes have divided into 2 because that's DNA replication. And then they line up in the middle in the metaphase. And then there's these microtubules that pull the chromosomes to the end. And in telophase, they reform the nucleus. And every cell then gets the same number of chromosomes. Here's the little diagram of it. Here's 4 chromosomes. And in us, it would be 90, it'll be 46 chromosomes, 23 from the mother, 23 from the father, but they all line up, and you get 46 chromosomes going to the in the diploid cells. Here's what the two bodies look like. They are made out of microtubules, and they are also related to the basal body of the flagellum. The sperm that moved towards the egg had a flagellum. That's how it moved. And at the base of it was the basal body, and there's another one there. So these basal bodies came into the egg from the sperm. Not only did the nucleus come in, but these things came in as well. So I went after the notion that the centriole and the 9 and 2 structure that Lynn Sagan and now Lynn Margulis had conjectured. I went after it. And the answer is that the endosymbiotic theory, that means the swallowing of a bacterium, of the origin of the centriole is found lacking, as no DNA has been found in the structure. The centriole is related to the ribosome in that it is a structure nucleated by RNA. This RNA, morphic RNA, has the property of localizing itself at the site of the old centriole before nucleating a new one. The implication of this property of localization of RNA is discussed with respect to the way the eukaryotic cell determines its shape. I sent this thing to Crick, and here's this lovely letter. He sends me back. It's a whole letter, which is magnificent. He says, "Thank you for sending your paper to Centriole. I cannot say at this time I'm very concerned about the evolutionary origin of centrioles, but I'm keenly interested in what they're made of and how they behave." This is what I've been trying to tell you. The molecular biologists were not interested in evolution. So this is a paper that I wrote in 1984, which is the origin of the eukaryotic cell. Where did the eukaryotic cell come from? It was endosymbiotic, and the central the point was the centriole and the basal body do not have a double membrane. RNA is implicated. Finally, the eukaryotic cell uses RNA in ways quite unlike bacteria. So basically this meant that the eukaryotic cell was something different than a bacterium. Now, what's missing is the chapter on Mendel, because now I have to talk about molecular biology. So the real question now is how, since I don't think that the centriole is coming from a endosymbiosis, how do I know that the mitochondria and the chloroplasts were bacteria? The answer is that each has a small ribosomal RNA that is bacterial in nature. There are other criteria such as DNA and double membranes, but it's now clear that the small ribosomal RNA is useful in identifying bacteria. In fact, it was Carl Woese that used small ribosomal RNA to separate the archaea from the bacteria and write a new world tree. We're now going to go to a tree that is no longer Haeckel tree. Now, I just have to tell you what you're missing. Why did Woese pick a small ribosomal RNA and use it now to identify bacteria, archaea, and also eukaryotes? And the answer quite simply is this, that every cell on Earth has a small ribosomal RNA because it belongs to the translational apparatus, which tells you how you read messenger RNA into proteins and how synthetases, tRNA synthetases, put the amino acid on tRNA, and how the code is invented right here on the, and how the peptide is made from the messenger RNA. Just to give you a little figure of it, here's the small RNA, a small ribosomal RNA, this is the large ribosomal RNA. Here are the tRNAs. Here's the tRNA with an amino acid on it. Here is the anticodon on the tRNA, and you're reading this thing, reading it 3 bases at a time, 3 nucleotides at a time. And this is what gives rise to the genetic code. Here is what the small ribosomal RNA, these are all nucleotides. It's about, this is an E. coli small ribosomal RNA. It's got helices, it's got loops, it's got whatever. And it was this sequence and these loops and these structures that were used by Carl Woese to give us the famous phylogenetic tree of Carl Woese. This is now bacteria on, this is bacteria on the right in blue. And then there is this interesting relationship between archaea and eukarya. And so we're going back to the eukaryotic cell. And now we're going to ask the question, where did the nucleus come from? And where did the centrioles come from? We agree that mitochondria and chloroplasts are endosymbionts. Well, here's the hypothesis, and here's my answer to the answer of why there's so much DNA, but not enough protein. A stands, it's the ABC hypothesis. A stands for archaea. That's the prokaryote that Carl Woese found on the basis of the small RNA. B stands for bacteria, and these are the results of the endosymbionts that became the mitochondria and the chloroplast. And C stands for the chronocyte, and in Woese's term, the progenote, the host RNA-based cell. Cronus was a god that swallowed his progeny. Cronus learned from Gaia and Uranus that he was destined to be overcome by his sons, and just as he had overthrown his father. As a result, although he sired the gods, Demeter, Hestia, Hera, Hades, and Poseidon by Rhea, he devoured them all as soon as they were born to prevent the prophecy. When the 6th child, Zeus, was born, Rhea sought Gaia to devise a plan to save them and eventually get retribution on Cronus for his acts against his father. What they did is they put in a rock, and he swallowed the rock, and Zeus was not devoured, and Zeus then caused him to regurgitate all his progeny. Now, this means that the host cell was an RNA-based organism and not a DNA-based organism. All the DNA came into the eukaryotic cell originally from archaea. Archaea was the endosymbiont that became the nucleus. And the centrioles and the are the remnants of the RNA chronocyte. So now it brings me to the end of my talk, pretty much. The next point is what's universal to the archaea, the bacteria, and the progenome. And that is the genetic code. And that has to do with what I showed you earlier. This is the genetic code as it's being read in the translational apparatus. Here are the alphabet. Here we're into written language. Here is the alphabet, A, U, G, C. These are the nucleotides. These are the amino acids. And then there are 20 of them. And these are the amino acids with hydrophobic side chains. That means they don't like water. There's those with the hydrophilic side chains that like water, and those that are basically neutral. Here is the genetic code as written by Crick. And as you can see, the way you read it, this is the first letter of the codon. This is the second letter of the codon. This is the third letter of the codon. Why are there 3 letters? Well, if there was only 1 letter, there would only be 4 amino acids. If there were 2 letters, there would only be 16 amino acids. 3 letters, you have enough for 20 amino acids. And that's why 3 letters gives you 64 codons. And now it's the mapping of those codons onto the amino acids. That's the genetic code. So on New Year's Eve, on a night of 1971, I rewrote the code. And this is just rearranging it. Instead of UCAG, I just rewrote it as GCAU and horizontal. And this is the first code, letter of the code. This is the second letter of the code. And this is the third letter of the code. And to make a long story short, what immediately came to mind was, holy moly, this is the simplest amino acid. These are simpler amino acids. Arginine is a bit of a problem. Here are the hydrophilics, and here are the hydrophobics, except for serine. And so here is the thing. This thing grew exactly the way ontogeny recapitulates phylogeny. It is terminal addition. That is, the code was evolved, and it was evolved as just as my embryology, early stages of my embryology. That was the key. That day, that night, I considered I solved the problem of the origin of life. And then this is what I wrote. You can read it at your leisure. And now the next question is, where do the amino acids and nucleotides come from? Where does the alphabet come from? And that's when I went to, since I was a biochemist, I went to metabolism. And as you could see, there's a citric acid cycle in the middle and everything else comes off of it, including the amino acids. Here's the citric acid cycle. And here's what I wrote. If we look at intermediate metabolism, in particular enambolism, that is the growth of the alphabet, we are immediately struck by the central role played by the citric acid cycle. In figure 1, which is what I just showed you, we see the anabolic functions of the citric acid cycle. If we apply the method of Bernal, which is the onion-like growth of something, it can be conjectured that the citric acid cycle came first, was followed by the amino acid, lipids, nucleotides, and carbohydrates. From a more detailed examination of metabolism, it is evident that some amino acids require fewer steps in their biosynthesis in the citric acid cycle. And then I just say glycine came from glyoxylate, etc. And then I said the summary is that to make the citric acid cycle and all the rest is that an autotrophic origin of life. That means it came from CO2, nitrogen, and H2S and phosphate, etc. And it requires clays, transition state metals, disulfide, dithiols, UV, and cyanide. A general scheme is proposed involving the fixation of CO2 and nitrogen for the evolution of intermediary metabolism based on the evolution of a complex system from a simple one. The basic conclusion is that metabolism could evolve from a simple environment rather than from a complex environment. The autotrophic, and it turns out that this going from the citric acid cycle, making the amino acids, correlates with how they came into the genetic code. Finally, I would like to read to you this thing in general. I can't read the whole thing because somehow I was just wondering if I can't get the full screen.
I can read it for you.
All right, why don't you read it for me, Jörg?
Yeah. Photosynthesis and the origin of life, Hyman Hartman. Abstract. The origin and evolution of photosynthesis is considered to be the key to the origin of life. This eliminates the need for a soup, as the synthesis of bioorganics are to come from the fixation of carbon dioxide and nitrogen. No soup, then no RNA will, no protein will. Cyanobacteria have been formed by the horizontal transfer of green sulfur bacterial photoreaction center genes by means of a plasmid into a purple photosynthetic bacterium. The fixation of carbon dioxide is considered to have evolved from the reductive dicarboxylic acid cycle, chloroflexus, which was then followed by a reductive tricarboxylic acid cycle, chlorobium, and finally by the reductive pentose phosphate cycle, Kelvin cycle. The origin of life is considered to have occurred in a hot spring on the outgassing early Earth. The first organisms were self-replicating iron-rich clays, which fix carbon dioxide into oxalic and other dicarboxylic acids. The system of replicating clays and their metabolic phenotype then evolved into the sulfur-rich region of the hot spring, acquiring the ability to fix nitrogen. Finally, phosphate was incorporated into the evolving system, which allowed the synthesis of nucleotides and phospholipids. If biosynthesis recapitulates biopoiesis, then the synthesis of amino acids preceded the synthesis of purine and pyrimidine bases. Furthermore, the polymerization of amino acid phytoesters into polypeptides preceded the direct polymerization of amino acid esters by polynucleotides. Thus, the origin and evolution of the genetic code is a late development and records the takeover of the clay by RNA. Stop.
Okay, so I'm going to end up in purely in a poetic note. The ultimate ancestor are these iron-rich replicating clays. This scenario that has just been read to you is a simple fact that our origin and the ultimate ancestors are these iron-rich clays which evolved into the cells that gave rise to us. In a certain sense, this is sort of going back to an ancient way of looking at life and its origins on this planet. It is this. It is going back to the old Aristotelian fire, air, water, earth, and how shall I say it? Creative fire. And so what it is The earth is our mother. The sun is our father. And what is happening is, driven by light, the water and the air are gradually evolving into us. And so it is that our ultimate ancestor really is our Mother Earth. And so I'd like to say we should honor her and should at least try to keep her going so that we can continue to exist on this planet. That's it.
Fantastic, fantastic. Thank you very much, Hyman, for a fabulous presentation. I think time is running out. And anyone has any quick question? All understood.
Andy, do you have any questions?
Sure. What are you going to do in a week?
I want to advertise. You want him to advertise his next talk, huh?
That's right.
The reason I'm not discussing clays, which is that I've just given Yuk a paper that I just published a day before I came to California. And in there is my whole history with clays and the origin of life, especially section 7.
Right. But he was asking, what are you going to talk about in the next lecture?
Oh, in the next lecture, it's basically, that's going to be theory. And I'm going to talk about von Neumann and the computer model of the origin of life, and that life and evolution is learning. So the next talk is the dominant person that I'm going to be talking about is von Neumann.
Very good, very good. And you're going to tell us the difference between the two giants, namely von Neumann and Alan Turing.
No, there is no collision between them.
No difference? Okay, very good. Very
Anyways, you'll have to wait till next week.
good. Okay. Okay, looking forward to next week. And thank you very much.
Okay,
Okay, bye.
Bye-bye.
bye-bye.
Thank you.
Anyways, that was the one thing that caused me some problem. That's why I asked you to read it.
Dr. Hartman, I have a question actually,
Sure.
an interesting question. How confident are we that all the ancestors of the life on the earth originate from the same planet?
You're not. Here's the reason why we haven't solved the problem of the origin of life.
Oh, I see.
There is a problem. That is, our planet is 4.5 billion years old.
Yeah.
We have records. So this is like history. I'm trying to do history just by reading the records. I'm not trying to invent a theory, except for the end. But everything I did was try to show you how there were records in us and in rocks and stuff like that. Yeah. So it's a historical thing. It's like, how do you know about a Chinese dynasty? Well, because you have records you can read. Yeah. So it's a historical thing. I was not discussing theory. I see. Until the end. Until then, it's just that here's a record. Here's the genetic code. Here's the record of the genetic code, how it grew. Here is metabolism. Here's how it grew. Yeah. That's not much of a theory. I mean, that's just say. But next week it is going to be all theory. I see. I really look forward to that. So this is purely a historical thing. It's trying to point out that just doing historical thing, you can go very, very deep. Yeah. Origin of the genetic code, origin of metabolism, and you can postulate clays. Okay. So that is theory. Now the reason is when you go back in time, there is missing. There's no records on Earth from 4.5 billion to 3.9 or 4 billion. Yeah. There's no rock record. Yeah. Life occurred in that region. Yeah. The earliest evidence for life is maybe 3.8. Not very good, but 3.8. Yeah. So the problem is that's where we don't have any records. Yeah. So then you could say, well, then you have to say, well, why don't we have records? Well, because rocks are constantly being destroyed. Yeah. Yeah. So the problem now is what happened in that 4.5 years of Earth. We don't have any records. Some records on the moon, but they don't help us very much, other than bombardment. I see. So then the question is, let's go to Mars. Yeah. Because Mars does have that record. It's not lost because Mars doesn't turn over. Yes. And maybe on Mars we can see what happened on Earth. Yeah. Yeah. That's why looking for life on Mars, origin of life, is trying to compensate for the fact we don't have records. Yeah. So the answer to the origin of life is, did life occur on Mars in the same period that it may have occurred on Earth? Yeah. Yeah. Or if you are crazy like Kirschvink, you believe that it was founded on Mars and came here as a meteorite. Yeah, that's very interesting. Anyways, but you can read my paper on clay. Oh, sure. And clays and the origin of life. Since it was, I mean, this chapter, there was a missing chapter about Mendel and the evolution of molecular biology, and all the work that I've done on evolution of ribosomes. And I mean, there's a whole thing. I'm taking those papers that I've written and putting them in groups so that if people want to read about some of the things I just talked about, they're welcome. Okay, thank you. So it worked okay? Yeah, it was great.